Welcome to BWB TV. I'm Cheryl Scott, Senior Technical Editor of Bioprocess International, and I'm sitting with Dr. Roger Nassar, CEO and founder of RAND Biotechnologies. I think your company spun out of MIT a couple years ago, so maybe you could tell me a little bit about the company and what it does.
Absolutely. Thanks, Cheryl, for having me. RAND Biotechnologies was established in 2012, basically following a PhD in fluorine chemistry and postdoctoralships. I had a couple of positions in industry, one at a large company, one at a small company. And then based on that experience, I founded RAND Biotechnologies in 2012 based on postdoctoral work in the chemistry department at MIT. Initially, during that first stage, in the first couple of years, we were mentored by MIT VMS, MIT Venture Mentoring Service. And we were also part of another incubator in the Boston area called the North Shore Ventures, and it's based in Beverly. Basically, those two organizations helped us launch our company, RAND Biotechnologies. They helped us from many different aspects. They helped us in terms of mentoring, how to access markets, how to identify markets, access them, recruiting customers, increasing sales. So both organizations were very critical at the start of RAND Biotech. Since 2012, we've been growing organically, mostly organically, recruiting customers, adding to these customers, getting them to have repeat business with us. And since then, we've been really on an incline. And we envision that incline to be maintained into the future.
Okay. What are the main technologies that you're working with?
Yeah. So if I want to summarize RAND Biotechnologies in a few words, I'd say we work at the chemistry-biology interface. Particularly, we make smart nanomaterials that have applications in biotechnology. So the common denominator here is that we make smart nanomaterials even the same materials could have different applications in biology. That being said, we focus on two main market segments. The first one, we focus on microfluidics, particularly droplet microfluidic markets. So we make materials for them to encapsulate biologicals for next-gen sequencing, for drug discovery, so on and so forth. We also make materials for capturing and detecting microbes. So we have a new technology to rapidly capture and rapidly detect live microbes.
How are those two technologies related to one another?
They are related at the nanoscale. They are related at the material scale. Okay. So based on our experience in various domains in the synthetic chemistry, we can functionalize materials in a way or another, the same core materials, but functionalized in that way or this way in order to accommodate their use in specific biotechnological applications.
Okay. So I hear that you can detect live microbes in a much faster way than what we are doing with environmental control and that kind of thing. So maybe you could tell me how that works.
Absolutely. We're extremely excited about our new technology. And we look at it as a very different way to look and detect live microbes. It's a very different way compared to traditional ways. So initial users, they look at our technology and they say, wow, we haven't thought that this could be done this way. It's a very cool technology. So in essence, we focus on detecting live microbes, live bacteria, fungi, and spore formers in samples. And what we focus on is we try to make it a true point of use technology that is fast and easy, and it could be applied in terms of the crowd here in the bioprocessing crowd. It could be applied at many different pain points in terms of the bioprocessing, manufacturing, metrology kind of setups.
Absolutely.
Now, and the way it works is that our smart nanomaterials, we have designed them to recognize and capture, like I said, live microbes. So I have here a device that helps us understand the technology. So all what the user has to do basically is mix a sample to be tested with our nanomaterials that come preloaded into such small tubes.
Okay.
They are inserted into a device that we have developed in-house.
Is that a smartphone that it's attached to?
It's a smartphone, and it could be attached to a tablet as well. It could be attached to a laptop, to a device basically that is capable of holding a small image recognition software. Okay. So this is where the third part of our technology come into game. So the first part, the essential part, is the smart nanomaterials that go after microbes, capture them to detect them. The second part is this small device, and the third part is the image recognition software. So basically our materials go microscopically after microbes, and they aggregate them, and they form at the end of the day a mass, 3D mass. Now the presence of this mass indicates the presence of microbes, live microbes to be more exact. And their size as well indicates how many cells we started with in the sample.
Right.
So the image recognition software helps us quantify that size of mass in the sample.
That would be based on the rate of replication.
Yes,
yeah,
absolutely. It is related to that. So our rapid microdetection, quick capture, rapid microdetection technology is a three-pronged technology. It's materials, device, software, and everything could fit in the pocket.
In your pocket, pretty much.
Yeah.
What do you see as the applications in the markets are here and maybe elsewhere?
Yeah, Cheryl, they're really limitless. I mean, for the bioprocessing crowd, as I mentioned, to detect sterility in bioreactors, in connections to bioreactors, samples, instead of sending them to a microbiology lab, they can be tested there and then where the sample is collected. And we have actually many studies proving that examining a sample, the microbiology composition of a sample at the site of sampling is extremely critical, as opposed to collecting a sample and sending it somewhere else. You lose lots of biological information.
True.
But then, because it's that simple, and it could be really operated by anybody.
Right.
You don't have to have a scientific training. It's a very short protocol, 6-7 steps, and anybody can use it. We had high school students using it. It's very simple to use. So you can envision now stepping out of the lab, trying it at home.
Right.
Checking for microbial contamination in the kitchen, in the bathroom, your water, drinking water, recreational setups, military setups, even on planes, on trains, detecting microbes not just in solutions but in air indirectly, and on surfaces, including fruits, vegetables, meat, so on and so forth. So it's really, the application markets are limitless for this technology.
What's the time? I know it takes a few hours at least, if not days, with a traditional taking samples and sending them off. So what's the time of the assay?
Absolutely. So we shrank down this time to minutes to hours. So it is very much related to the live microbes. So they need to be alive, and we pride in the fact that our technology differentiates between dead and live. Okay. And yes, we shrank it to a few minutes to a few hours.
Oh, that's wonderful. Yeah. So when will we be able to see it on the market? I'm sure a lot of companies are going to be really wanting to give it a try.
Yeah, absolutely. We're currently ready.
Yeah.
So we do have kits, and these kits include our materials and the device, and we're ready. We've been sampling it to early adopters that have been testing samples anywhere between a few hundreds of microliters to 1 milliliter. And we also have customers, actually early adopters, that have been testing it with half a liter. So that's another major advantage to our technology. It's not limited by the size of the sample.
Really?
It can be on the orders of half a liter, if not bigger.
So you have a little cartridge for this one, but you have different models basically that handle a larger and larger sample.
Absolutely, absolutely. And we take advantage of the fact that our nanomaterials are insoluble in aqueous environments, so we can at any time separate them. So these could be mixed with big bulks of samples, liters even of samples. And then at the end of the day, we look at our materials and try to see if there are microbes in there or not.
Right.
So we are ready. We've been supplying it to early adopters, and now we are in the process of recruiting more early adopters and initial customers to try our materials.
Do you have a booth here that people can find you at?
Not today, but we have a presentation tomorrow, the round robin table panel presentation, and I'd be happy to meet with more people. And of course, in the future, look us up. We are biotechnologies.com. Connect with us through our website. We'd be happy to answer any questions and happy to ship to you guys kits to test.
Awesome. Fascinating technology. Nice to meet you.
Thank you, Cheryl.
Thank you so much for coming here.
Thank you.